Hey guys, so the topic here is going to be DNA structure. So I'm going to just do a basic run through on the structure of DNA and nucleic acids. So I think you guys should have talked about nucleic acids and monomers and polymers when you talked about biochemistry at the beginning of the year, but I'll kind of just do a quick run through on that too. So the four different macromolecules are carbs, carbohydrates, fats, these are your lipids, protein, and then nucleic acids. So the carbs, proteins, and the nucleic acids, they all have something that's known as a monomer and a polymer unit. All right, mono means one, poly means many. So for carbs, proteins, and nucleic acids, they have a small individual unit for each of them. In the case of carbohydrates, it's called a monosaccharide. Proteins are amino acids, and nucleic acids are nucleotides. They all then have a polymer unit, where if you were to take multiple of the monomers of carbs, or multiple of the monomers of proteins, or multiple of the monomers of nucleic acids, and you made a chain of them, that would just be called your polymer unit. So the polymer unit of carbohydrates is not monosaccharide, but polysaccharide. The monomer unit of protein is not amino acid, the monomer unit of protein is amino acid. The polymer would then be a polypeptide or protein. And then the nucleic acids, which we're going to elaborate here, are called nucleotides. A nucleotide, that is the monomer unit of a nucleic acid. And the structure of that is that it starts with a little sugar, and that sugar is called deoxyribose, hence the term deoxyribonucleic acid. That's what DNA stands for. This is a deoxyribose sugar. This is called a phosphate group. I'm not going to get into depth on what the actual chemical structures are. This is just called the sugar. This is called the phosphate group. And then over here, you have something called the nitrogenous base. It's made out of nitrogen, so it's nitrogenous. But we can just call it the nitrogen base because it's a little easier. Okay, so this is the structure of every nucleotide. It's going to be like that. Phosphate's going to be the same. The sugar is going to be the same, but this is going to change a little bit. Okay, this is going to determine what type of nucleotide you have here. So there are four different types of bases you could have. You could have adenine, thymine, cytosine, and guanine. So these are just abbreviated A, T, C, and G. If you were to put an adenine base right there, you would have an adenine nucleotide. You could put a thymine right here and you'd have a thymine nucleotide, and so on. That's just how that would continue. Okay, so if you have one of these, one of them, that's going to be a monomer. It's just the one nucleotide. If you were to put a bunch of them together, it's going to form a chain. You're going to have this long chain of nucleotides. Okay, that's going to be your one long chain. If you were to get another chain of them, put that together, I didn't do that exactly right, but you'll get the picture once I draw this. It's going to create your, like that, your squiggle, your double helix. So I'll make it look a little nicer like this. Like that. That's what it would look like. And there's your double helix. So this side is just one long chain of these attached to each other, and the other side is just another long chain of these put together. So that's where that structure comes from. The structure that was discovered by Watson and Crick who used Rosalind Franklin's picture. She took the picture. It was the cross section was like right in here. So that picture that you guys looked at was like this cross. So they were able to determine that the helix twists around itself from her picture, and that's what they won the Nobel Prize for. And if you saw in that video, she didn't not win the prize because she was a woman and they were all men. She died of ovarian cancer, most likely because of her studies with using x-rays to study this stuff. So that's why she didn't get it. Okay, so just a basic run through on DNA structure. Please let me know if you have questions on this.